Okay, everybody, we are on Module 4B, Lab 8, and we started our lab early because we have, we're working with our semi-permeable membrane, okay? So inside the semi-permeable membrane, you have a starch solution. Outside, you have a cup of water with iodine, all right? So the idea is that some things will be able to go through that semi-permeable membrane, and some things will not be able to go through it. Okay, so by the time we get to end of class, you should see a change taking place. I like to give it more time. That's why we start at the beginning. All right, so here, this thing, did you have to write any of these things down? Okay, cell membranes protect the cell. They keep the pressure of the cell stable, and they have special proteins allowing things in and out. It's semi-permeable. It makes a decision if something gets to come in or doesn't get to come in. All right, so the plasma membrane looks something like this. All right, it provides structure, support, and regulation of substances. So this interesting picture, you can see there's water at the top, water at the bottom. Water going through the semi-permeable membrane is pretty good. I mean, water's small, it's H2O, it's a small molecule, and it'll fit through there without much trouble at all. So if a cell needs water inside of it, the water will travel in by osmosis automatically. Okay, it'll just travel in there. But there are some things that are too big to go through there, and they have to have a special channel or tunnel or something to take them in. All right? We call this the fluid mosaic model, and you can see right here the unique structure of the plasma membrane has a bilayer of phospholipids. We call it the phospholipid bilayer. It's kind of like a sandwich. Okay, we have a piece of bread and a piece of bread, and they come together. It comprises the whole membrane, not just around the cell itself, but usually around the organelles as well, inside those eukaryotic cells. And you can see here this little ball at the top is, so this is outside the cell on the top here, this is inside the cell on the bottom. We have a ball with two little legs sticking down from it. I'm going to show you a picture of it a little closer up in just a minute. And what happens is that the top part, the little red ball, is called hydrophilic. It likes water. Water will dissolve in that. But the little tails are hydrophobic. Water doesn't dissolve in there. But guess what will? Fats will. All right, so fats, it's a beautiful design to allow water-soluble and water-insoluble things to get in the cell if needed. So it looks kind of like this, right? We see the little orange dots here with the little legs coming down. So our hydrophilic and our hydrophobic areas, like the little sandwich. And this purple thing is called a protein channel, and it codes for certain proteins that fit in it, and they will allow those to come through. Proteins are kind of big compared to like water and ions and things, but even ions sometimes need to have a special, even a protein carrier that takes them in through it, a little tunnel, a little channel. And sometimes the channel has a charge on it, like a positive charge will draw a negatively charged ion through. Question?
Did we learn about this last week? Because I feel like we did look at something like this.
We looked at this when we talked about the plasma membrane in general. Now we're getting a little more specific. Okay, so we had this picture last week too, right? Permeability. Remember see how big those red blood cells are, those big round red things? They can't fit through those little holes that look like Swiss cheese right there. They can't fit through, so they won't go through. Only some things will travel through, these little purple and yellow and green balls, but not the red cells. Substances can move through the plasma membrane, which is selectively permeable. That's what you're writing in that blank, selectively permeable. Small molecules can pass through easily, but the membrane does not allow other larger molecules to pass through. And in this particular picture right here, this is good news, because we don't necessarily want blood traveling outside of our blood vessels. We want to keep them in the blood vessels. Bless you. So in this particular case, we don't want the blood going into the outer tissue there. We want the blood staying in the blood vessel. But we want the nutrients from the blood to get to the tissues. That's why we have stuff circulating in our body, to get oxygen and CO2 and nutrients from our lunch. Okay, so here's that little picture to the right here. We have the little ball. See, it's a hydrophilic. That means it loves water. And the tail is hydrophobic, meaning it hates water, it fears water. And therefore, we could have fats would dissolve in this part, but waters would dissolve in that part. So hydrophilic means water loving, hydrophobic means water hating, or technically water fearing. Phospholipid is a lipid where one of the fatty acids has been replaced with a phosphate group molecule. That's just biochemistry right there. It's important for the structure of the plasma membrane, but I don't think it's important for a quiz. I don't think you have a blank
It
on
looks
that.
like the ends of these things are burning.
Ah, there's something changing inside there. Yeah. Yeah, the cups show your little tube changing. All right, transport types. We have passive transport. That's like water going in. Water will go in to a cell if the cell needs water. Just automatically go in. That's passive. It'll passively go through. But sometimes I have to use energy to get things in. That's called active transport. And the energy that I need is called ATP, adenosine triphosphate. All right, so if I asked you what's the difference between passive and active transport, you would say?
One needs energy and one doesn't.
Yeah, which one needs energy?
The active.
Active. All right, so we can see right here, here's some of those channel proteins. And there it looks like water is going through there very easily, yes. Here's our phospholipid bilayer. All right, and they're just coming through. And it talks about it being our solvent. Water's the universal solvent, right? Stuff dissolves in there easily. Okay, so it's passing through very easily. So passive transport includes things called diffusion and osmosis. Diffusion is the random motion of molecules from an area of high concentration to an area of low concentration until it equals out, it evens out. Osmosis is the diffusion of water through a semi-permeable membrane from an area of high water concentration, which is a low solute, to a low water concentration. Are you all set to write that whole thing down?
Yeah, I just shortened it down.
It's good. That's good to learn how to shorten because in college they don't really wait for you.
I wrote something else down, so I had to erase it. I wrote simple diffusion and then facilitated diffusion.
Facilitated? Is that how it says in your book?
Well, yeah, I was looking in the book and it had two different types. It had osmosis, simple, and facilitated diffusion.
Okay. So, but both of these notes are passive, right? We don't need energy for this. It'll happen, but it's what we call a gradient. For instance, in your tube right now, what was inside your tube?
Starch.
Remember? Starch, a starch solution. And we know what happens to starch when it gets in contact with iodine. What does it do?
Turns black.
Turns
I
black.
say green. I
Yeah.
say green.
And sometimes it looks a little purple, actually. And remember, these don't require any ATP. No extra energy is required. It just goes against the gradient. All right? All right, so the types of solution. Concentration turns out to be important. All right, the first one is called a hypotonic solution. It's a solution that has a low solute concentration relative to another solution. I'm going to show you pictures of these in a little bit. It's going to really help explain it better without using all that scientific jargon. The next one's hypertonic. Hypo means beneath, below.
What?
Okay. Hypo.
Really?
Yes. Yes, if I have hypothyroidism, I have
Oh,
too
so
low of a production of hypothyroid. Hypothyria is to below the temperature of your body. Hyper means above. Hyper is above. So hypertonic, the solution has a high solute concentration relative to another one. So low, below, high, above. Okay, that's hyper. If someone's hyperactive, they're way up here. Woo! If they're hypoactive, they're like kind of laid back, right? That's me.
I have two big words that I can use now.
Yeah, all right, good. And then isotonic means it's even. Solution has the same solute concentration relative to another solution. In the human body and living things, because we are talking about biology here, in living things they like to be in an isotonic environment. They don't want it too too stressful by being too high of a solute inside, outside. They want everything nice and even. Remember, we call that homeostasis. We want to achieve homeostasis. So I'm going to show you what happens to cells in these cases, okay? Oh, this is an experiment in your book. Yes, in your textbook, okay? And according to this, it's a required deal. But we're doing this today in your cups right there. So I'm okay if you don't do this. But if you do this at home, the reason why we can't do it in class is because it takes time. It takes too much time. And therefore, we just don't have the time to do it. So what you're going to do, you can read the instructions. Essentially, what you're going to do is take an egg, 2 eggs, put it in a cup of vinegar overnight, which dissolves the shell off the egg, but leaving the membrane intact. All right, so then you have, the bouncy egg. Y'all have seen that before, right? The bouncy egg? Yeah. So you have that, and the membrane around it is a semi-permeable membrane. It's pretty cool. All right, so you're going to take that thing and you'll put it into a cup of water, and the water will start to head into it. And then the other cup is into a cup of high fructose corn syrup, or Karo syrup. And I would guess with all y'all, you'd probably have to go buy that, because most of the moms, some moms are healthy moms, right? They probably don't have high fructose corn syrup in here. In fact, we avoid high fructose corn syrup in anything that y'all eat. Oops.
We don't get syrup.
Yeah. So this is if you've ever had those peanut butter cornflake cookies. You ever had those that are so good? They're made with high fructose corns. I mean, you literally get the Karo syrup. Or a popcorn ball is coming on Halloween here. Have y'all ever got a popcorn ball for Halloween? It's made with that stuff. It's kind of chewy, very sweet, and we've decided it's very bad for us. It's not good for us. Oh, it's so good. It's peanut butter and, bless you, Karo syrup and butter, and it's cornflakes.
Don't think you ever had this.
Oh my goodness, it's so good. All right, so then you're going to see how you're going to end up with one of them getting hypertonic and one of them getting hypotonic. Okay? So if you do that, take a picture of it, show it to me, I'll give you extra credit. All right, now active transport and passive transport. Active transport is the movement of substances through a plasma membrane that requires energy. It is not osmosis or diffusion. Passive transport is movement of substances in or out of the cell due to osmosis or diffusion, not requiring any ATP. Keep in mind, I should have put that on active transport, requires energy in the form of ATP, adenosine triphosphate. Okay, so if I have an isotonic solution, see this picture over here, y'all? I have just as much water going in as going out. Everything's perfect. Everything's in good shape. It's isotonic, right? It is maintaining homeostasis. This, by the way, up here is an animal cell called a red blood cell, and below is a plant cell, okay? We're getting the same amount of water in as out. So what can happen to a cell when the solution is not isotonic? Well, we can have trouble if there's that happens. So bad news for cells. Hypertonic solution is a solution in which the concentration is more than the cell that resides in the solution. So we see here what happens is too much water goes out of the cell. All right? So hypertonic solutions result in something called plasmolysis. Plasmolysis, the collapse of a walled cell's cytoplasm due to lack of H2O, lack of water. It is a dehydration of the cell. So it results in plasmolysis, cell dehydration. That's what you're supposed to write, I think. What's the result of a hypertonic solution? Plasmolysis, which equals cell dehydration. They get all shriveled like that. And we all know that dehydration's bad, right? It's not good for us. We want to be hydrated all the time. So that's a hypertonic solution. This is, for instance, if I have a, let me think of it, if I say it right. If I have a freshwater fish and put him in a salt water in the ocean, he'll get all shriveled like that. Yeah. His water will go out of him. More bad news for cells, a hypotonic solution, a solution in which the concentration is less than the cell resides. It's less than the cell, so what happens is water's going to go in, go in, go in. A hypotonic solution can result in cytolysis, which will rupture the cell due to excess internal pressure. You're right. Cytolysis, cell rupture. So that's taking my saltwater fish and putting it in fresh water, they'll explode. Boom. Now it doesn't pertain to some particular fish out there called, what's the name? Anadromous. Anadromous. We'll get to those when we talk about fish, like salmon, for instance.
It actually just bloat a lot.
Yeah, but it can kill them.
Yeah, oh, yeah, yeah.
Yeah,
die.
yeah, it kills them.
They don't,
right.
like,
They don't
explode.
actually explode. It's
Yeah.
kind of like if we go into space without a spacesuit, we just kind of pop like a piece of popcorn. Yep. Yeah, like that. All right, so isotonic, everything's good. Hypotonic, we have cell rupture from too much water going in. Hypertonic, we have cell dehydration. They get all shriveled up. Okay. Passive transport, also called facilitated diffusion. I believe, David, that's what you mentioned earlier. Facilitated diffusion can have help by facilitated diffusion. Okay, so passive transport can be helped. Facilitated diffusion is the movement of certain molecules across a cell membrane through protein channels, like we saw in that little animation of the water going through those little channels. And they go from an area of high concentration to low concentration. That's just a natural, if I got very little water inside the cell and there's more water outside the cell, it will go in automatically, right? It'll get in because the cell wants more water in there. So we have that facilitated diffusion. You can diffuse in this way, but we can do it faster if we have something like this. And this one to the right we'll get to has to do with active. That little deal on the right there is active transfusion.
Look at mine.
Okay, what substances move this way? What substances do it at? Ions, sugars, and salts. Ions, sugars, and salts.
Purple.
Those are all very small, although some sugars are pretty big. All right, and then we see active transport then requires ATP. That's your next blank, ATP. Maybe speed it up a little bit.
Yeah, I think it's because you tied it off really well.
Shouldn't make a difference.
Really?
The tie has nothing to do with it.
Oh, it's dissolving the spices in it.
I think it just needed a more concentrated solution of iodine. Okay, large molecules or molecules moving against the concentration gradient need help of a transport protein. Y'all write that down, a transport protein. This experiment we're doing right now is the reason I always carry string with me. I leave it in there because trying to do this experiment, I didn't have any string.
How did you do it?
We didn't get to do it.
Oh.
Yeah, so that's why I've always, instead of trying to remember to put it on, I keep it in that bag at all times. Okay, types of active transport. Okay, this is trying to get large things into or out of cells. Endocytosis, particles into the cell with vesicles. Endo, particles going in. Exo, particles going out. Exo, getting them out. And then a big one, phagocytosis. The word phago means having to do with eating. Okay? So phagocytosis is the idea of cell eating. They're grabbing large chunks and pulling it into the cell. So you could even say cells engulf large particles or other cells, for that matter. The best example of this in the human body would be white blood cells engulfing bacteria. And then, of course, neutralizing it. Yeah, y'all's is looking really good over here, you two. And Riley, yours is
Sad.
a little slower. Kai, yours is doing all right? Looks pretty good, yeah. All right, so we're going to take a look at these three things individually. Watch a little video on them. Oh, goodness. Okay. All right, here's our phospholipid bilayer. Remember I said like a sandwich, here's one piece of bread, here's the other piece of bread. So phagocytic vesicles or vacuoles form when the plasma membrane will engulf it. This whole thing will come around and engulf it. Pinocytic vesicles form when the cell absorbs large molecules but smaller than the phagocytic ones. And secretion vesicles, the cell might be making something inside the cell and go to the Golgi body and then release it. Yeah? Remember the Golgi body releases synthesized materials? That's a secretion vesicle. I don't think you're writing any of this down, are you?
No, I don't think so.
Yeah, I think I added this in because I thought it was important information. Okay, so now pinocytosis, also known as cell drinking. Cells take in fluids, mostly fluids like this, and they'll pinch in like this, you see, and then pinch off, and we have a little vesicle now with that stuff inside there that's going to now be useful to the cell in some way. So the cell takes in fluid containing dissolved solutes like sugars or proteins. It's sometimes called it's called pinocytosis, sometimes called cell drinking. Let's watch a little video on it.
Pinocytosis, often referred to as cell drinking, is a way for a cell to take in substances that are too large to travel through the plasma membrane. When the cell encounters such a substance, it actually breaks its plasma membrane, allowing the substance into the cell. A pinocytic vesicle then forms around the substance so that it can be transported to wherever the cell needs it.
Okay, so we're just taking kind of, it's too large to go through regularly like water or something, but it's not huge, so it can go through pinocytosis. Phagocytosis, the cell takes in a larger particle, let's say a solid if you will, a large protein. This is also known as cell eating, so notice we have a large thing in there. Let's watch the video on this one.
This microscopic video
That's a
shows a
white cell.
white blood cell attacking a yeast cell. The particular species of yeast here is Candida albicans, which is a pathogen, so the white blood cell is eliminating it. This particular white blood cell is called a macrophage, and it eliminates pathogens by phagocytosis. In phagocytosis, the cell simply engulfs what it is trying to attack. As you can see in the video, the white blood cell has engulfed the yeast cell, and it will now simply digest the yeast cell to get rid of it. White blood cells, like this macrophage, engage in phagocytosis in order to protect the body. Other cells simply engage in phagocytosis to eat. Amoeba, for example, engulf their prey so that they can eat them.
Okay, yeah, so the white blood cell has that kind of amoeba-like movement, and it surrounds the pathogen and gets rid of it, all right? And so he called it a macrophage, so big eater, phage, to eat, right? Big eater. So pinocytosis, taking in the solutes, the fluids mostly. Phagocytosis is taking in larger substances, bacteria, things like that. Okay, secretion, remember the cell's making something. For instance, the cells of the pancreas are making insulin. So it takes that insulin and it's going to give it out to the rest of the body so the rest of the body can use the insulin, and the insulin's job is to take glucose into your muscles so you can move, okay? So it's very important. So it's going to take it, it's going to fold it. Remember, it gets some stuff from the endoplasmic reticulum, then it folds it up and it modifies it. Then it sends it out in a little vesicle here so that it can go to where it needs to be. So notice how it makes that little vesicle around it. Look how it's covered completely in a phospholipid bilayer itself. And another little video here.
When a cell needs to secrete something for use by other cells, the Golgi bodies put that substance into a bulge, and the bulge begins to grow until it breaks off, becoming a secretion vesicle. The secretion vesicle then travels to the plasma membrane, causing a breach in the membrane so that its substance can be released into the surroundings. The substance can then be used by other cells.
All right, there we go. Review, check for understanding. We have hypertonic solutions, hypotonic solutions, and isotonic solutions. Which ones do we want to maintain in our bodies? Isotonic, right. That is maintaining our homeostasis.
Hypo's too low, hyper's too high.
Exactly. Yeah, too much, too little. And then we're going to see, oh, here it is. Yeah. Plasmolysis, shrinking, right? Too much, shrinking. Hypotonic, too little, it bursts. Isotonic, concentration is stable. So maybe put that little hint down there for yourself. So when you go study for this for the quiz in 2 weeks, no, next week, right?
Yeah.
Quiz is next week? Yeah, I would know that if I were you. Hint. Put a little star by that one. So passive transport, here we go, depends on the concentration gradient. So we can have passive transport through diffusion, osmosis, or facilitated diffusion. That's using a little channel of some sort. Active transport, we can have endocytosis, taking it into the cell. Exocytosis, getting it out of the cell through these two processes of phagocytosis or pinocytosis. Phagocytosis is the larger things, pinocytosis is smaller, but still too big to go through on a diffusion mechanism. summarizing, active and passive. For active, we've got to have, what is it?
Water?
ATP.
Oh, ATP.
We have to have ATP. So see that little door? That little door is going to have to like open and close. So here's an example. Here's the little door. Here comes my ATP. It's going to cause it to change. And now my other thing is my ATP is going to come back in right here. Get that door moving. Notice that phosphate attaches to it, and that phosphate is going to drop off, attach to that ADP, make a new ATP, so we can start the process over. What we're looking at right here is something, a very, very complex structure in our body called the sodium-potassium pump. This is what's happening every time a message is being sent through your nerves from your brain. All right, and like right now it's happening because y'all are watching something with your eyes, and your brain therefore is telling you what you're looking at. And when you do something like that, this all has to happen. We have to get the sodium and potassium, which are two positively charged ions, back into the inside or outside of the cell, depending. All right, here's another one. Here we're going to have something that fits perfectly right in here, that little H thing, whatever that is. And notice it kind of went kind of quickly. The ATP is going to come in, it's going to devolve, and then allow that to open up and release it into there. And the phosphate goes to get that phosphate group gets recycled. All right, time for lab. All right, yes.
I wonder what we're
We
gonna
gotta go
do.
back, huh?
What is ADP?
ADP is adenosine diphosphate. Did I not have that anywhere? Okay.
Everyone knows that.
So adenosine triphosphate has 3 phosphates. Why don't I have that on there? I'm sure I put it in there. I put it in like 5 times. Okay, we're going to be making a model of a cell for our lab, including, in addition to what you're doing there in the cup, we're going to make a model for our cell. And our cell, remember, looks like this. Okay?
Are we drawing that?
No. Is there a place to draw that?
Yeah.
No, on your lab sheet? No. We're going to do a special lab right now with this. Okay, and then, oh, my mouse is dead.